we should probably kick things off. And so the first presentation today will be from John on the OpenMM project. I turn it over to you, John.
Thanks. I'm super excited to be talking about OpenMM, and most of the folks here are actually most of the folks listed as contributors here, especially Peter, who's been the developer since the start, are actually at this meeting, except for Gianni and Jaime, who couldn't make it today. And so you can ask them technical questions if you want to. So I'll tell you a little bit about some of our work on trying to leverage the enormous structural investment of structural data in the PDB. And it's been actually remarkable how rapidly the structural information we have access to has been growing. Just in the last decade alone, there's been 100,000 new structures. And so making use of this information for drug discovery, for biomolecular modeling to understand the origin of disease has been certainly a question that a lot of us struggle with. We tend to use these physical models. Often they're called molecular mechanics potentials. They include a representation of all of the nuclei in the system, and then use a simple algebraic expression to model the interactions between them in a way that's rapidly computable. There might be force fields like amber, charm, and amoeba that you've heard of before. They have various levels of detail, modeling things like multipolar moments or other interactions. But the basic essential ingredients are that they're physically motivated and try to represent the overall energetics and forces in the system. And these kinds of models allow you to answer a whole number of questions about the molecular mechanism of some biological process at the biomolecular level, or how a physical perturbation to the environment or to a mutation, via mutation, or the binding of an exogenous agent can cause disease, how you might design a small molecule or biologic to restore normal function and cure that disease, then how you might modify things to change their properties, or assessing the impact of human genetic variation or variation in cancers, for example, on whether a drug will still work or not. So OpenMM is a package that's intended to be a very flexible way of answering questions like this by building interesting algorithms on top of it. The website has a very easy to use navigable interface to all the documentation and user guides and documents. And I'll tell you a little bit about what the package does. It's basically several layers that intend to make it as easy as possible for developing or using molecular modeling types of physical modeling abilities in whatever application you want, or if you're a direct user doing the modeling yourself. So it starts with the highest or the lowest level code is all in CUDA and OpenCL, and it's easy to add new kinds of things for other architectures as they come along to take advantage of hardware acceleration on consumer or commercial level GPUs. There's a C++ API that allows it, that is very modular, so you can easily extend it with plugins of your own if you'd like to. And then it's all wrapped in an interface for Python and other language bindings, so you can use it from a variety of different languages. And only a little bit of the code is at the highest level to make it as easy as possible to use in whatever applications you want. But we're providing more and more of the Python layer later on, so you can easily compose that into higher level algorithms. So there's a number of different applications people have built either in our group and many other groups. I'll give you two quick examples. Many of them are at the Python level simply because it's so easy to put together applications there. We've also been building things like OpenMM tools, which allow you to encapsulate a lot of otherwise complicated algorithms, again, with a nice level of abstraction to build things on them. And then again, the core is in C++, but can be used in a variety of different language bindings. Just to give you one quick example, this is Isolde, built by Tristan Kroll at Cambridge University. He's just using OpenMM as a way of doing real-time dynamics with restraints to the electron density to do interactive real-time modeling. And in this case, he's rethreading a helix that was mismodeled by the structural biologists. And you can do all sorts of amazing things by easily augmenting this with additional forces and then running on the GPU as you're doing other kinds of things. Another example from our own work is that we've been working on this collaborative open science drug discovery project for COVID-19 to develop an orally bioavailable therapy. We just learned that we have oral bioavailability in rats today, which is really exciting because that means we're really close to clinical trials. But we're using OpenMM under the hood on Fulling at Home to run very large scale, tens of thousands of free energy calculations to tell us which small molecules will bind better. This is just a small layer of code on top of OpenMM itself. One of the really powerful things about modern machine learning frameworks is that they provide this really nice level of abstraction where you can easily define what you want to do by having really good abstract objects that allow you to essentially execute your workflow very simply. So we're trying to do the same kind of thing here for molecular simulation, this little script, actually built by a script builder that's online for first-time users, allows you to pick a structure and load a force field that's used to assign the energetics, define the simulation you want to run, tell it which GPU you want to run on, and then do your basic operations like minimize the energy, equilibrate the system, say what you want to save from a simulation, and then run that simulation. And instead of having its own scripting language, which a lot of other molecular mechanics codes do, it's just Python, and it interoperates with the entire Python ecosystem, making it very flexible. We've tried to make it as easy to use, as easy to use as a library, as well as an end application as possible. We spent a lot of time working with the CondaForge folks to deliver OpenMM with CUDA toolkit on any platform so that it automatically can use CUDA or OpenCL on whatever platform you install it on. And so we're really excited to finally be deploying this. It's in a testing channel right now, but will be deployed after another week of testing to the main CondaForge channel. Folks all around the world use it, and we've been lucky enough to have hundreds of thousands of downloads so far. There's a very active issue tracker where people ask all sorts of questions, and we get a lot of insight about how we can make it more usable from folks. It's very fast and works pretty well on inexpensive consumer-grade GPUs, so you don't need Tesla cards or anything like that. You can use off-the-shelf consumer-grade gaming GPUs, and we're able to deliver less than $1 to get a microsecond per day, which is pretty cool. And it's also run on an extraordinary amount of hardware. So over the last few months, we were lucky enough for Folding at Home, which has been supporting a lot of experimental collaborators and uses OpenMM under the hood for their GPU compute core to grow to a ridiculous number of users. Over 1 million people have been running OpenMM over the up to May, for example, and we passed the exaflops threshold, mostly powered by OpenMM. And what's cool is that we've been able to benchmark and assess how well OpenMM works, and this is the previous version of OpenMM that's used in that core, on a super wide array of hardware. So it's been extremely well tested on many different devices. And we're able to use that information to both give people an idea of what the efficiency is on different kinds of hardware, and also to make sure we work well on a wide variety of hardware. It's not easy every day that you're sure you run on 1 million different pieces of hardware. It has a lot of built-in physical forces that encapsulate some of the stuff you might use in standard molecular modeling, and we're always adding more by working with folks who are working on the cutting edge. But it really allows you to also flexibly define algebraic expressions that automatically get differentiated and compiled for the GPU. And this was implemented actually many years ago, far before AutoDiff frameworks and machine learning frameworks became very popular. And Peter's intuition about how to make this super fast has been really super useful for making flexible prototyping software that still runs at GPU speed for the community. There's a bunch of other things that automatically reduce these to kernels for different kinds of interactions. If you want to model your own kinds of things externally, you can control a lot of them externally as well. And we're really trying to make this as easy, like our end goal is to end up with these kinds of abstractions that allow you to do things like build new force fields. This is with the open force field effort, by just defining something in a machine learning framework so that you can use this very familiar way of being highly productive to build and fit new force fields, for example, and then deploy them within things like OpenMM. We're also making it easy, like I said, through these other packages like OpenMM tools to compose really complicated algorithms. This is a whole free energy code that also does some parallel or some tempering of temperature in between. And this is all you need to be able to define things where you'll define an alchemical region to decouple from the system. You'll define your protocol for doing this calculation. You'll say, I want to actually add in some Monte Carlo displacements and rotations, and then use Langevin dynamics for the rest of the sampling, and then use this effective replica exchange algorithm to sample from everything. Now, the future of OpenMM is really interfacing better with machine learning frameworks. So Peter and others have put together both the PyTorch and TensorFlow plugins, and we're looking into how to support JAX so that if you define either a force, like that is something that computes the energy given a configuration of the system, or an integrator or a collective variable you want to use as a restraint in any of these machine learning packages, which are often much easier to program in than CUDA or OpenCL, you could then bring those in through this plugin and load in your model and deploy it within the simulations very easily. So that's becoming something that we'd like to have first-class support for. And we'd also like to go the other way and have machine learning potentials easily be used in these frameworks so that we can build new models, because we're seeing a revolutionizing of this field where using simple models like these ANI potentials, for example, which are quantum machine learning potentials, that is they use some simple basis functions, they push it through a deep learning network, and then they're able to compute things like quantum chemical energies with high accuracy to DFT or even coupled cluster at this point. It's more expensive than molecular mechanics, but it's orders of magnitude cheaper than quantum chemistry. So we think these things will supplant QMMM, where you'd otherwise use quantum chemistry, very expensive quantum chemistry for parts of the system, to deliver much higher accuracy. And this is super exciting because the real near-term application is modeling ligand-protein interactions for high-level or for high-accuracy drug discovery, where you really dial in on the accuracy of your ligand and its interactions with the protein by replacing part of that with these machine learning potentials. My group has recently put out a preprint where it shows that we can cut the error in predicted binding affinities, for example, for ligand design, where you have this congeneric series, in half, which is a huge increase in accuracy. It really reduces the number of compounds that you would need to synthesize to actually make active molecules. So this is going to be an active frontier of research. You could even simulate the whole system with these quantum machine learning methods. And this is really interesting because it makes a lot of calculations, like computing free energy differences between different tautomeric states or other ligand designs, very simple. We're working on making that efficient, of course, but the cool thing is that these can learn not only from quantum chemistry, but also from experimental data. And we haven't been able to have our machine learning or our molecular mechanics potentials do that before this. So you can actually train, for example, in this case, on tautomer free energy differences. And then using that experimental information, you can generalize more broadly from a limited set of data. So we're excited to support applications like this, partly funded by the Chan Zuckerberg grant over the next few months. Soon, you'll be able to do entire simulations of entire proteins. This is really slow right now, though. So a lot of the community focus has been on how do we make this fast enough? And that's something that OpenMM intends to really accelerate by building GPU custom kernels. And there's a variety of different models people have used and are interested in exploring. The basic idea is that they have some sort of bottleneck, either in the neural network computation or often in the feature computation, that just isn't handled very well by these machine learning frameworks, where these potentials are implemented right now. So what we're hoping to do is to use this opportunity to build a community-driven library that encodes GPU-accelerated and CPU-accelerated kernels that can then be used either in the machine learning frameworks or in MD packages like OpenMM. And so we've already broken ground on that and are starting to integrate that in. We also need some way to deploy these models to make them as easy for people to use. And so we're looking into building a machine learning model repository that'll make it very easy to package and deliver these once you've built a great model that you'd like other people to use. So with that, I'm just going to say we're hoping to also provide quantum chemical data that you can use to build better models. There's already this thing that we've helped support called the QC Archive that's run by the Molecular Sciences Software Institute and partly supported by the Open Force Field Initiative. And we're hoping to use the CPU power that we've recently found on Folding at Home to add more quantum chemical data to this, to really flesh out this archive of publicly downloadable quantum chemistry data sets that you can use to improve these biomolecular force fields for various scenarios. We think that'll provide a nice ecosystem for the OpenMM users and folks in structural biology. So with that, I want to thank CZI for funding us as part of this last cycle. It's been super helpful to keep the project going and to embrace new things like machine learning, and to all the folks who have contributed. I couldn't possibly list them all here, but happy to answer any questions.
So, the past couple of weeks, as someone maybe not deep in the structural biology field, I saw the headlines about AlphaFold 2, and I actually saw there was a tweet indicating that OpenMM was used in some of the refinement.
It was.
So I would love it if you could maybe share a little bit about that and how you see some that intersection with like structure prediction and the usage there.
AlphaFold has been really exciting. Of course, it showed up 2 years ago, and DeepMind was working on the previous CASP blind prediction challenge. They're trying to predict crystal structures before they're released to the public. And one of the big advances has been using these evolutionary couplings. So sequence information across different orthologous or homologous sequences has been used to define which parts of the protein should be in contact. So they predict these contact maps and then use them with a machine learned potential, essentially, to kind of build the structure with the right details, and then load it into things like OpenMM to do physical modeling, where it's very hard to simulate an entire protein folding, for example, with the physical model, but we think the details are right enough that it really helps you refine those last details of those. And OpenMM is one of the few packages that's just publicly available and has an API for molecular simulation, so that's why they probably picked it. It's also quite fast for the GPUs that they have available.
So exciting. Yeah, thanks for sharing that. Curious if we've got a couple of minutes, if there are any questions from anyone else. Lucas.
Yeah, so I have a very basic question because I don't know that much about molecular modeling, but it's just I hear a lot about from people around my own laboratory here. They are using something called GROMACS quite a lot. And how do OpenMM relate to GROMACS?
They're quite different. GROMACS is one of these, everything is included kinds of packages that has a very vibrant ecosystem as well. BioExcel is the main lead developing entity behind that. It's kind of a closed system where there's lots of packages, lots of bits of it that interoperate, and it was not designed with an API in mind. That they're trying to bolt one on in recent years. So they're very different philosophies, and I think we've been working closely with the OpenMM developers to try to come up with portable ways to define a system and take it from one to the other, where GROMACS excels at CPU performance with very highly optimized assembly codes. OpenMM excels at GPU performance, and they're really built with different things. GROMACS is great for end users, and OpenMM, but it's harder to use as a library, and then OpenMM is really easy to incorporate as a library.
So following up on that, I mean, I guess I think of when I've heard of, and I don't work in this area directly, but when I've heard about GROMACS, I think of it as doing a lot of people use it for molecular dynamics kinds of things. Is OpenMM also in that space or only on the MCMC side of things?
Yeah, you can definitely use OpenMM for molecular dynamics. That's probably the major use of it. But you can also build much more complicated integrators and other kinds of things into it if you're doing much more elaborate kinds of calculations or algorithmics. So, for example, to implement a different kind of integrator because you're interested in particular properties or want to take very large time steps, it's a very long odyssey where you have to send someone to sit with Eric Lindahl and learn the internals of GROMACS for a few months to be able to implement that into GROMACS, whereas it's a few lines of code to implement a custom integrator in OpenMM for molecular dynamics. So it really depends on what kinds of things you're doing. If you're doing the things that GROMACS is already built for, it's great. But if you need to extend it, then it's an odyssey because it's really designed for full optimization on CPUs, and anything else is a little bit more difficult to implement.
So this is a follow-up question. I have a biophysics PhD who's doing a postdoc in my lab and developing his own MD engine based on CBMC. And should he be making it compatible with OpenMM in some way?
Sorry, CDMC?
Yeah, CBMC is a kind of
configurational bias Monte Carlo. Yes.
Yeah.
It might be very easy for him to implement it in terms of layers on top of OpenMM, in which case you'd get access to all of the potentials and all of the other things that we have integrating with OpenMM. It would be much harder to do that with GROMACS, I think, because you'd have to call out to the shell every time. But he could easily build it as Python layers on top of OpenMM. Might be something he looks at.
He's already got it, and it's like directly on the GPU. So the question is whether if we should make this, if we want to, is there some way we could encourage adoption by getting it to be offered as part of OpenMM?
That's an interesting question. We might set up a time to chat, but I think it's a more complicated thing because you would have to share information on the GPU already to get access to the other kinds of capabilities. It's definitely possible, and there might be a way to write a plugin that moves information back and forth efficiently. Okay. But it would be great to follow up.
Okay.
So I have a very, very technical thing. You mentioned that you have Python bindings, right? And I think, so you're doing C or C++ mostly in your kernels, or maybe even assembly. So we have a similar problem in OpenMS, and I was wondering if you have the same. So sometimes I'd like to write a C++ interface, and I'm not allowed to do it because Python just cannot handle it. So I have to write extra code just to make Python happy, which usually doesn't make me happy. So did you encounter something similar, and do you have a solution to that?
Peter might be able to speak to this, but my experience certainly has been that it's difficult to come up with good, robust Python bindings. I think PyBind 11 has been the favorite of the community so far. We're using something that's a little bit embarrassing, I'm afraid, but Peter can speak to this.
Yeah, well, we use Swig to generate them. And Swig, it's amazingly powerful. It can do just about anything. And it can sometimes be very, very painful to figure out how to get it to do that. So I wouldn't say it's an ideal system, but it does work. And with enough effort and sweat and pulling of hair, we've always been able to get it to do what we need to do.
Great. With that, let's move on, Chris, to your talk. And thanks again, John, for that great presentation. The whole OpenMM team, really exciting to hear about that one.
Okay, perfect. So this talk is going to be about next generation mass spectrometry with OpenMS. Yeah. So my name is Chris. I work at the Bioinformatics Solution Center here at Free University in Berlin. And so yeah, let's get rolling. I hope. Let's see. Yeah. Okay. So just as a attribution, basically. So we got this CZI grant, but I didn't write the grant, right? That was Hannes. He's currently heading a lab at the University of Toronto. Also, Timo from Oliver Kohlbacher's lab had a say in this grant. So they both basically contributed and they got the money basically. So I'm just here to talk about the results, right? Because we actually then got into it and got to implement some of the things that the grant actually aimed for, right? So okay, so that's that. Now. I wasn't sure about the audience, so I'm going to spend a few minutes on a brief introduction to mass spectrometry. I know there's quite a few people here who know a lot about mass spectrometry. You might want to cover your ears because this is going to be very brief and rough. And I was hoping to give an introduction to everybody, right? So I'm going to misuse some terms on purpose, I'm afraid. Please don't be mad. Okay, so the setting that we're in here is basically we want to analyze biological data, yeah, and what we usually have is some kind of biological sample, and we're interested in the proteome, right? So we're doing proteomics and high throughput proteomics, and so what you want in the end is you want to quantify and identify as many proteins as possible, yeah, with the goal in the end of using that as a kind of biomarker, for example, right, to treat diseases or to come up with new chemicals, basically. And then the method of choice in proteomics is mass spectrometry, right? Also abbreviated MS, and that's also where the name of our software, OpenMS, basically is derived from. And so what you do is you extract proteins from a sample. You digest that into peptides. So basically, you're making a problem more complicated by splitting your proteins into tinier subunits called peptides. And since now you have a very complex sample, what you need to do is you need to make it less complex in order for a mass spectrometer to basically handle the data. And the method of choice here is chromatography. So you're adding an extra dimension, which in this case is called retention time. This is in the order of minutes to hours. So let's say 10 minutes to 2 hours is usually the length that you would run a chromatography, and then whatever goes through that. So there's a small column which is used. Proteins or peptides in this case, they will be retented to a certain based on their properties basically, and they will come out at the other end, not one by one, but that's the rough idea, right? So you have and that's the basic point where you basically couple that to a mass spectrometer, where you can now acquire mass over charge scans, right? So the next dimension that you're going to acquire is mass over charge. On the y-axis you see intensity, yeah, and this is basically what this $500,000 instrument will do for you, right? So it's basically a very expensive scale, yeah. Now you can acquire many of these mass spectra as the data flows out of the chromatography, and in the end, if you stick them together, you can make nice 3D pictures, right? This is now done also with OpenMS, and now you have 3D data, and that's where the bioinformatics comes in, right? And just to give you a feeling about the order of magnitude that we're talking about, so depending on the type of instrument and how you look at the data, we're talking 0.5 gigabyte to roughly 90 gigabytes per run, right? So for a 2-hour gradient, you could acquire like 100 gigabyte data, right? And you need ways to figure out like how to process this, right? So it's not, it's probably not gonna run on your mobile phone. You need something better, right? But OpenMS tries to invent algorithms which run on many platforms, right? Now the tricky part here is the configuration of the mass spectrometer, because there's many ways how you can acquire this data, and in what way you acquire this data, and then this has influence on what algorithms you should be using to analyze it. So to acquire one such mass spectrum, one widely used acquisition mode is so-called data-dependent acquisition, DDA in short. So what you do is you acquire a simple mass spectrum, which basically gives you, at a certain time point in the chromatography, all the peptides that are basically eluting at that time point. And you're looking at the intact peptides, basically, and each of those peptides is going to give you a signal in the M/Z dimension, and the intensity is going to correspond to the abundance in your original sample to a certain degree. And now what the mass spectrometer will do, as it's acquiring this survey scan, as it's called, it will pick the n highest signals. I just picked 4 here. Most mass spectrometers these days can do 15 or 20, but for the sake of the example here, let's assume it's 4, right? So you pick the 4 highest signals, and then you're trying to isolate this signal and analyze it further in a so-called fragmentation scan. So all you know at this point is the mass of the peptide, which might not be unique and not tell you what peptide this actually is. So what you want to know is the sequence, and you get this by doing a fragmentation on this peptide, and luckily peptides fragment very predictably. So machine learning could come into play to analyze this. We do it a bit differently, but nevertheless, if you analyze this, the signal here, fragment the underlying peptide, you might be able to assign a sequence to it, and this will help you in identification, right? So the usual data flow here is you do a survey scan, MS1 scan as it's called, then you do multiple MS2 scans, usually of the highest ones, and then you repeat this process over and over until your chromatography, is running out, basically, for 2 hours or something. And so you acquire up to many hundred thousands of spectra in one run. And so the problem here that you can immediately see is whatever n I pick, there might still be signals which I cannot identify, right? So there's still some things which I'm going to miss. And that's actually a problem, because this technique now is biased towards the high abundance peptides, right? So you're not going to see or be able to identify low abundance things. And it's a bit stochastic as well. And I'm skipping over a few details here, but that's basically the story, right? Then back in 2012, some guys at the ETH Zurich came up with the idea that, we could actually do something else. We could change the way the mass spectrometer is analyzing our data. So coming back to this MS1 scan that you see here, what you're doing is you're not picking, the highest peaks and then forgetting about the smaller ones. What you actually do is you're using wide isolation windows, and for each of those isolation windows, which they call SWATH windows, for sequential window acquisition of all theoretical mass spectra. That's the abbreviation. So basically you fragment everything that you see, and it's data independent. That's why it's called DIA, because you don't care what data you actually see. You just fragment everything. That will take a bit longer to do, basically, but that's also not a problem with modern instruments. And what this allows you to do post hoc is basically you have all the data there. It's just a bit more, difficult to look at it, because now, as you can see, these windows are a bit wider as they were before. Before you would just analyze the single peak, roughly speaking. Now you have a wider isolation window. So if you're a bit unlucky, you observe more than one peptide signal in this window, and you need to figure out, you need to disentangle this a bit, right? But basically, so you're running these MS2 SWATH scans, and when you're scanning basically over your whole mass range, you just start all over again. And when you repeat the cycle, basically the information from the chromatography will have changed slightly, but this will still allow you to basically reconstruct a chromatogram, which you can see on the right here. And this you can basically use if you know what you're looking for, right? This you can basically use to identify peptides, right? And so this is the acquisition mode that is, new and the next generation of mass spectrometry, as Hannes called it, right? It has certain advantages and certain disadvantages, so you cannot do this with every instrument, but this is very popular these days to do. Okay, so that's basically the data that we're working with. So now coming to OpenMS, because that's now the software, right? So you acquired the data. The question is, what do you do with it now? Well, one answer could be you just use OpenMS, which we have been developing for like the last almost 20 years now. So it's open source BSD3 clause. I just looked it up. It's 500,000 lines of code. Now, someone said a dumb programmer writes a lot of code. Smart programmers write less code. So I don't know where this is, right? But just to get a feeling, this is 500K lines of code. We have many tools in there, and it's also a library. And one of these tools that we have is called OpenSWATH, which is used to analyze this kind of data, that I just described. Okay, now the rough OpenSWATH pipeline looks like this. It's at first it's a bit overwhelming, but it's actually not that complicated. So you have this data independent acquisition, raw data that I just described, right, coming right from the instrument. You feed that into our tool, basically, along with basically a dictionary, a spectral library, as we call it, which tells you, well, what proteins and peptides do I know, and where do I need to look for them, right? So you need a theoretical idea of what proteins are out there, and how in the data, shall I identify them? So you need to have a theoretical model, and the OpenSWATH algorithm will just extract the data and give you candidates, basically features of which it doesn't really know which ones are true, but it gives you all kinds of data to work with to decide that. And once you've done that, you get chromatographic information, like where it found them. And then you can pass this on to another tool, which is written in Python, hence PyProfit. And it will make some statistical analysis of this, and we'll assign Q values, which you can use to basically decide which ones of those candidates are actually true. And then you can even go further down and analyze multiple samples together. So you get a comparison across samples and make some further statistical analysis. And what you get out is basically a quantitative matrix, which you could almost stick into Excel. Well, we don't use Excel, right? But that's the point where you are at this stage. And so basically, where we tap in now with our CZI grant is to improve the usability of this workflow. And the first idea was we use a wizard to run this pipeline basically for new users, because if you do this on a command line, it could be a bit troublesome. Also, you don't really know where to put what data, right? And so we just wrote a wizard to do that. So that's basically concentrating on the highlighted part of the pipeline, right? So it subsumes all this and puts it into a nice GUI. And the second part is basically data visualization. So you basically want to know, what did I identify, and is it actually any good? So you can browse the data. So there's going to be a tree structure, so from proteins to peptides and feature groups, and you can just look at this tree and look at the data and the annotation, and then decide if the algorithm did a good job or not. And then there's a third part, which I'm not going to talk about. So imagine you have thousands of runs. You probably want to analyze them all, and you cannot even do it in a local cluster. So you want to go cloud, right? And that's another part that was done here. But I have no information actually what happens, so I'm not going to talk about it. So coming back to this usability with the wizard. So what you want is to run the workflow locally on a machine and just see what comes out. So what we did is we wrote a new tool and we call that SWATH Wizard. We added roughly 5,000 lines of code to that, much of which was just, Boy Scout rule, leave the code in a better state than you found it. So there was a lot of refactoring coming into this, and we're using Qt to do to do the visualization, and it's now part of OpenMS, and you can just look at this. And this is basically how it looks like, right? So I'm just going to show you a few screenshots. So you basically have a few tabs where you can just input your data via drag and drop, and it's all neatly described, and it looks at, is Python installed? Do you have the correct modules and stuff? And if not, it tells you where to go and how to do it. And then you can load your libraries, your assay libraries, and it tells you how much is in there, like how much can you actually find in your data if you're really lucky. And then, it basically goes on, there's a few parameters which you can set. And then, you just click a run button, and then you're golden, right? That's the hope. And if you're really interested in it, there's a log window where you can copy all the gory details and re-implement the workflow in like a workflow engine, whatever you like, basically, right? So you can use Nextflow or something else. Okay, so, and the second goal that we had is, so basically to visualize all this data that OpenSWATH annotated, so you can inspect the features that it identifies and browse the proteins and peptides. Also there is roughly 6,000 lines that we added, some of which we removed also in Qt, also available as part of OpenMS now. And so basically what we did is OpenSWATH tends to use SQL, to be more precise, SQLite, because we found this to be a really performant way of storing large amounts of small data. And it works reasonably well, and I'm sure you can even improve on that. But for our purposes, it's actually not too bad. So we're talking almost 1 million extracted ion chromatograms, and it can handle that quite easily in like 500 megabytes of data. That's not too bad. And on the annotation side, you have 8,000 proteins in your library, which translates to something like 100,000 precursors. So it's peptide with a charge, basically, and a couple of features which are annotated. And this also goes reasonably well. The schema is a bit more complicated, but it works really nicely. And so if you now load both of these kinds of data into our viewing module, basically, which is called Top View, you can basically look at all the extracted ion chromatograms. So this is similar to looking at a mass spectrum, but it's just a different dimension. And you basically get all the features that were annotated, and the one that wins is now shown in green. And ideally, it would be on this green dashed line because that's where the library says it should be. But chromatography is not perfect, so it's a bit off, but that's totally fine, right? You get a Q value, and you can basically, on the right-hand side, you browse your peptides and proteins. And then you can basically decide, are my settings correct or not? Obviously, right, if you're into machine learning, you would say like, can we automate this? And surely you can and you should. But nevertheless, people in the wet lab, they always want to look at the actual data. So it's a good confirmation for them that actually the right thing happened here, right? And you can basically go through this. If you want to try this yourself, OpenMS, right, you just go to GitHub, you install it, and you just run the tools. Yeah, and that's it. Next up, if you can find the time, we want to analyze, because 3D is not enough, right? So there's 4D data looming around these days, which has ion mobility dimension added to it. So that's something that's going to be next, and the data is even bigger and a bit more challenging. And then something we haven't come around is GUI testing. So if someone of you has a good idea, please, let me know how to do that properly in C++. Okay, this is the reference for the original publication for SWATH, and then some sources which I stole from Wikipedia. They are, referenced here if you want to look at this. And that's basically the end of my talk. Thank you very much.
Thanks. This is really great work. We've been doing some work where we're trying to do probabilistic inference from experimental data, but one of the problems we run into is that the vendor formats are all proprietary and totally different from each other. Is this standardized in the mass spec community, the output file formats, or how did you solve that problem?
Yeah, to a certain degree. So we are a bit lucky that we have the Proteomics Standard Initiative, PSI, and they came up with a good format, which is called MZML. Well, good in quotes. So basically, what it does is every vendor now has seen the necessity to be able to convert to this common format. The format itself is maybe debatable because it's XML and it's a bit bloaty. But nevertheless, we have this. There's this common format. There's software out there, MS Convert, from ProteoWizard, which can basically convert into this format, and basically OpenMS is just using this, right? So we don't bother to read vendor-specific formats. We could, because if you do, sometimes there's additional information in there which is not included in the common MZML standard, and sometimes that's a pity. But usually we get along just nicely, yeah. So that works rather okay.
That PSI sounds like an awesome success story. Does CZI think about helping nucleate initiatives like that?
Yeah, so we are having discussions around in the imaging space. There's a sort of plethora of file formats out there from various vendors. And so I'm always sort of interested in success stories. I made a little note of the Proteomics Standard Initiative there as well. And I think that's a lot where, one can learn across the different disciplines too. So Matt, you're up next.
So I'm Matt again. I was lucky to join the Olga Witek Lab thanks to the CZI funding. MSstats and Cardinal, Next Generation Statistical Mass Spectrometry in R was the title. Here I will be presenting just the MSstats part of the project. So I was lucky that Chris actually described the mass spec data analysis workflow. So I don't have to, and I will show you where MSstats lies in this workflow. So let's start with this, why MSstats needed, even before I describe MSstats, let's start with why MSstats needed some additional development. So the mass spec data is getting bigger. So that's more protein, more samples. That includes the DIA workflows that generate massive data compared to DDA, for example. Even TMT data that are a large focus of MSstats ecosystem packages are also bigger than label-free data. So we needed to make the data analysis, the statistics more efficient. Also, there are different processing tools that generate different formats. So we've seen one with OpenMS, but the number of different formats is much larger. So we needed to adjust a bit for the diversity of input formats that we can deal with. Additionally, we're going for helping in doing reproducible research, for updating our documentation, and also facilitating sharing data sets and analysis. So I will refer to that later. Moreover, we wanted to refactor or improve the MSstats to accommodate new experimental workflows. For example, extensions for PTM data, and to make MSstats ready and extensible for possible other new workflows. So in general, to facilitate new developments that can wait MSstats. And our efforts help in multiple ways. So first, we redesigned the package in a way that makes it very easy to connect MSstats with various signal processing tools or post-processors to create larger MS data analysis pipelines. I will also describe kind of an automated testing infrastructure that we needed to develop, which can also be used for testing, evaluating, benchmarking larger pipelines and finding the optimized pipelines consisting of signal processing, post-processing, statistical analysis. And also we developed some abstractions that make MSstats, like I said, very easy to connect to signal processing tool, but it's also a chance to create these larger pipelines where we can evaluate some choices, like the approaches to filtering shared peptides and so on, including the statistical part. And since the topic of the meeting is collaboration, I think all of these improvements are opportunity for collaboration. Okay, so let's start with this. What is MSstats? We've seen the large part of the flow of data in mass spec analysis, and MSstats is located somewhere at the end of this data flow. So it takes as an input to the output from the signal processing tools such as OpenMS and OpenSWATH. So it's kind of tool agnostic. It requires just a list of identified and quantified peaks. And from there, it performs statistical analysis to detect protein that change abundance across some conditions. And right now, it's actually a family of packages that, thanks to our refactory efforts, are really interconnected, share a lot of codes, and allow for possible new extensions. But the main package and the original package is MSstats, which performs significance analysis for DADA, DIA, or SRM data. MSstats TMT is a newer package designed specifically for TMT data that depends on MSstats for some parts of the analysis. And currently, both of these packages depend on a new MSstats convert package that abstracts the pre-processing data from signal processing tools to the format that is suitable for statistical analysis. And then other packages also depend on these main packages, like MSstats PTM, MSstats TMT PTM for statistical characterization of post-translational modifications. Also, for example, sample size calculator for classification. All packages are listed here, but these are the main ones, interesting ones. Plus, as a part of the MSstats ecosystem, there is a public data repository called Massive Quant, which allows sharing and reanalyzing MS datasets. So like I said, there's more packages in the ecosystem, but I will focus on the two main ones, or mostly the MSstats package, because the workflow that's in there is very similar to the other packages, and also on the new package, MSstats Convert, which we developed recently. So the general workflow in MSstats, like I said, is kind of tool agnostic. The input are just quantified and identified peaks. So we start with that. We do some data pre-processing, for example, normalization, to kind of fix data artifacts, some technical variation. Then, after data is processed this way, feature level data, so these peaks are quantified, meaning summarized to a single value per protein and run, or a protein and run and channel in case of TMT data. So we get protein level data that can be used for statistical modeling. It's done in a robust way that accounts for outliers and missing values, and it prepares the data for actual statistical comparison. So from statistical perspective, this methodology is very modular. So normalization, summarization, other steps are separated and come before actual statistical inference done for each protein. And as a part of the project, we also move the modularity to the implementation level in a way. So then after the data is summarized, quantified, we can detect proteins that change their abundance across conditions, and we've got some visualization to present the results. This is based on a family of flexible, linear, sometimes linear mixed models. And also the data are treated as a starting point for a follow-up study. So MSstats allows calculating power of a study or required number of biological replicates for classification or for a new study for statistical power. And let me show you what's new. So the new parts are a more general and modular interface that adds to the statistical modular more of a software modularity. So what we had before in MSstats was already high level of interoperability with other tools for mass spec data analysis in the form of converters. So a set of functions that allow to import data from various formats into a consistent format that is suitable for statistical analysis. So it satisfies requirements of MSstats, of statistical modeling for whatever tool we used. So we made it more general so that we have a common framework for the converters that already existed in the MSstats ecosystem, but also makes creating new converters for new versions of existing signal processing tools or for other processing tools that were not implemented yet very easy. So in a sense, we have a very small abstraction for multi-file inputs. Then the only thing that needs to be written for a new tool or version of a signal processing tool is a function that will combine possibly multiple files into a single table with the spectral peaks. And then we have a very general function that captures all pre-processing steps that can be required before we start statistical analysis. So that is filtering based on Q values, filtering specific peptides, aggregation of data, handling isotopic distributions, and so on. We also modularize the processing step in a way that makes it very simple for the user. Okay, so the old interface was such that all processing was done by a single function. Now we divided this into steps, which allows the user first to save intermediate analysis steps. And secondly, it allows users to use external implementations. So it's possible to mix implementations provided by MSstats with custom data processing steps and analysis steps. So for example, to achieve better performance, it is possible to run normalization separately and just pass normalized data to other functions. Or for example, for protein summarization, it is possible to use just a summarization function and parallelize the code in the most efficient way. That is data specific, infrastructure specific, and so on. Also, to add to reproducible research part and flexibility of our software, we introduced a new logging system based on the log4r package with a high degree of freedom for the user to save to existing file, new file, print messages, and all the logs. Each step of the analysis, it can be saved to a file and then they're checked for problems or for the processing steps that were done. Interesting part of the analysis was that while refactoring code and other new features, we had to test the code to make sure that it still works correctly. And it's actually hard or even impossible for unit tests that can be covered by, for example, continuous integration to cover all cases, because some cases are very subtle and data specific. So we tested all our updates, all the changes that we made on over 30 or rather close to 40 data sets which is very time consuming. While we were able to make our code more efficient, that, for example, recently a 7 GB DIA data set was processed in something like 3 minutes. It's very data specific. Sorry, it's very data specific. So some data sets may run for even hours, while some other data sets of same time, same type, for example, data sets, TI data sets, one can run in minutes, I can run in over an hour. So for 30 data sets, this can take really hours and hours and requires also manual work. So we also developed a testing infrastructure that's based on GitHub and Travis CI. So actually a free tool for continuous integration, which triggers a cloud build of our software, in our case, AWS, which then triggers a script that runs the checks that we require for each data set, despite them coming from different tools, and generates a report that we can check to see if there were any problems. Okay, so to wrap up, the tools can be found in different places. Firstly, Bioconductor for the stable versions of the packages. Then the GitHub VTech Lab organization for development versions. There is an official website with documentation and also a Google group for bug reports. Also, I provide a link to the Massive Quant tool for sharing data sets. We also organize training. The videos from May Institute and other short courses can be found on YouTube for May Institute or for Olga VTech Lab. And lastly, I want to acknowledge the development team. So Mina Choi, the lead developer, Ting Huang, the MSstats package, Tsung-Heng Tsai with the MSstats PTM package, and others. Thank you for your attention.
Great, thank you very much, Matt. Very enjoyable. We've got one question already in the chat from Bill, which I'll read out. And Bill asked, what file format do we need to produce so that you can list our Crux toolkit among your possible inputs?
Actually, it's just a matter of writing a new converter. So any example input from Crux is enough to produce a converter and add it to our set of predefined converters. And so the formats that we support out of the box.
Okay, maybe we can talk to you about how to do that. Thanks.
All right, we've got one more final presentation from Lucas. Would you like to take it away?
So I'm Lucas Schall, and I'm here to present the Percolator analysis engines for tandem mass spectrometry data. So I'm going to divide my time here into two parts. Firstly, I will give a background to shotgun proteomics and as well as to the Percolator algorithm. And subsequently, I will talk about recent improvements that we've done to the Percolator code base. So this first part will be pretty easy for me, as Chris and Mateusz already has sort of given the general outline of proteomics experiments. But the shotgun proteomics is currently the most comprehensive technique for analyzing protein contents in any biological mixture that involves cleaving proteins into short-length peptides, which you separate and inject into a mass spectrometer. And the mass spectrometer in turn, that produces spectra. And the spectra are used to identify peptides and proteins from. And here today, I mostly will talk about the peptide identification problem. That is how to go from spectra into peptides. And this is sort of a thing that we solve with, or most of the time we solve with something called a search engine. So a search engine is a piece of software that compares every collected spectrum to simulated spectrum from a target database, and the target database is just a sequence database, a FASTA database. And the result of this is matches between the spectra and peptides, and they are called peptide spectrum matches. Each peptide spectrum match is associated with a score, and you normally just keep the top scoring PSM or top scoring peptide spectrum match for every spectrum. So, as I said, PSM is the abbreviation you use for peptide spectrum match, and I will use that during the talk. This procedure is normally expanded as well with some extra stuff. You not just match it against a sequence database or a target database, you add extra sequences or nonsense sequences or decoy sequences, as they're known. These are non-feasible sequences, so any match against a decoy database are an example of an incorrect match. And as they are incorrect, we can count on them as being representative of other incorrect matches against the target database. And hence we can calculate statistics from observing these. So we can calculate things like false discovery rates, etc. Matches or PSMs stemming from the target database are called target PSMs, and PSMs stemming from the decoy database are called decoy PSMs for short. So Percolator was sort of built in response to a situation that was before we wrote the software. It was that quite a number of heuristic scoring schemes existed for what was to be called the correct match in the end of the experiment. So you would put up thresholds on the primary scores coming out from search engines, as well as using secondary scores and different peptide length features, etc. And we at the time realized that it was sort of made sense to combine these kind of features into one single score using machine learning technology. More specifically, we used a semi-supervised machine learning technique. We count all the decoy PSMs as being examples of incorrect matches, and while we, during the process, find out whether we can count on the target PSMs to be correct or not. So, Percolator is a so-called self-trainer. It's iterative in its procedure. It takes an initial scoring function and score target PSMs and seeing which of them pass a certain false discovery rate threshold. And we then use that as a training set for an SVM trainer, a linear support vector machine trainer, which we can then forward to a new classifier and making an even better selection of a positive training set. And we do so over and over again until we converge. So this procedure gives you not just now false discovery rates and other statistics, it as well increases the yield of the procedure out of shotgun proteomics. When Percolator was firstly published, we were producing almost 100% more PSMs per experiment, actually. Nowadays, the search engines are kept up, or have catched up a bit. So now we're getting more moderate increases. So here's an example from the draft human proteome, where we find about 15% more PSMs on a 1% false discovery rate. So Percolator been around for quite a while. We designed it firstly during 2006, 2007. It's published 2007. It was open source from the start, but it's been on GitHub since 2009. That's as well where it keeps the issue trackers, etc. And it's a C++ program. It contains approximately 8,500 semicolons, and it's been around for a while. And actually, we're so happy right now to receive this funding from CSI, because we've been maintaining Percolator on a shoestring budget for quite some time, and here we have some possibilities now to expand a little bit more on the software side of Percolator. So here are some recent modifications to Percolator code base. So firstly, we were able to introduce a continuous build system for Percolator. We shopped around a bit and selected GitHub Actions, as it's it was easy to generate artifacts from it. And so in our case, we generate binary packages directly for both Ubuntu, Mac OS X, and Windows for the system. And we can introduce a better testing framework. We've always been maintaining system level tests, but now as well are able to do things like unit testing with Google Tests, and as well introducing coverage tests with GCov. All these are things that we just couldn't do before, or just didn't have time to do before. Shotgun proteomics, as anything else inside of the business of biology, it's getting bigger and bigger. We are getting larger and larger data sets, and hence we are seeing need for speeding up the code a bit, because we're seeing larger and larger data sets, and the data processing become increasingly a bottleneck. So a couple of years ago, a couple of people, including Joel Halloran, were looking on different ways to improve the speed of Percolator by parallelizing this training procedure. At the time, they did so using POSIX threads, and we unfortunately couldn't really make use of that code because it didn't port well into all our platforms that we supported. But so now we were able to convert this to OpenMP, and hence we can now use it for Windows, etc. as well. It's this parallelization helps the overall speed dramatically. It sort of reduces it to a fifth of its original training speed, or a fourth, maybe more accurately. Another speed up that we're currently looking at is to use, well, Percolator is heavily depending on partitioning data into high or confident target PSMs using FDR thresholds. And this normally involves a full sorting of all peptide spectrum matches that we have. And however, and that's sort of time consuming when you get a large data set. So we found an interesting method to avoid full sorting and still being able to set full discovery rate thresholds using so-called layer order heap sorting. This reduces the full discovery rate calculation time by 60% or so. It's well, the procedure is more or less, it's sort of a way to sort PSMs so that you guarantee to do increasingly large windows or large layers, where each layer have elements that are guaranteed to be larger than the elements in the layer before it. And that amount allows us now to home in to which of these layers that contains the full discovery rate threshold that we're looking for. But this is still work in progress though. Another possibility for us now is that we can generalize the use of Percolator to other problems or new problems. Particularly, we've been looking on the possibility to use Percolator as an aid in performing protein quantification. When you are comparing different runs, you often find errors both in mass and retention time, and these can be weighted optimally using the Percolator algorithm. So this is something we have done in a recent publication, and we as well are thinking about extending this procedure for other types of for other quantitative search algorithms as well. So with that, I want to attribute to a couple of people, particularly people from Bill Noble's lab at the University of Washington. Brian Reiter and Charles Grant have been instrumental here for developing the test code inside of the project, and I as well want to thank Matthew Thay, who have been extremely and enormously helpful in supporting the Percolator code during the years. And as well, would like to thank Oliver Rang for introducing this thing with layer orders heap sorting for me, and John Halloran at UC Davis for working on the support vector machine training, and as well my own lab at KTH. So thank you for your attention.
John, do you maybe have a hand to raise? Yeah.
Yeah, I was super excited to see CZI getting more into structural biology, because I think the strength had been on imaging and then genomics in the past. So just wondering if you could say a few words about CZI's interest, or maybe I should wait till the ask about open science thing.
No, that's a good thing. So, at CZI, we have a number of areas, kind of domain-specific areas where we have existing grant programs. And we have a large effort to support the Human Cell Atlas, imaging. And then, we have an effort to support kind of open science more broadly. And this program, the kind of EOSS, was part of that open science program, and really looked to sort of support some of the foundational open source software across the different domains. I think there's a comment of structural, and proteomics as well. Proteomics might have been a much better word. Yeah, I'll pass that feedback along, and it is appreciated. And I think what we wanted there was to support both these kind of domain agnostic tools. You'll see that like parallel sessions right now where we've got NumPy and SciPy that are very cross-cutting, and then also the kind of foundational software in domains of importance to biomedical science. And so this kind of proteomic structural biology area was I think it's obviously a critical part of science, and one that we weren't doing much. And maybe I'll sort of speak from my sort of more personal view on this than like kind of representing the whole organization and its views, but personally, I see that it's a great way for us also to learn about what is the kind of cutting edge sort of software computational problems in domains that we aren't actually yet invested in. And so, I take away a lot of learnings from today and some of the other projects we've supported about what's happening in these domains.